learning objectives will be that at the end of the lecture, you will be able to appreciate that what are carcinogens, pro-carcinogens and promoters. You will also revisit normal growth regulatory genes. You will also know some of the examples of carcinogens, pro-carcinogens and promoters, types of damage inflicted by the above agents, genetic lesions caused by them, and what are the hallmarks of cancer due to carcinogenesis. The agents which are producing cancer, DNA is attacked by carcinogens. Directly it is attacked by carcinogens. The carcinogens may be chemical carcinogen, physical or biological carcinogen. DNA may also be affected by chronic inflammatory states like chronic infections, chronic inflammation, and DNA which is already a bit abnormal but hasn't produced cancer yet, and it runs sometime down in the family, and the person is said to be predisposed to these cancers. So when a second hit or second insult is inflicted on the DNA, whether it may be chemical, physiological or biological, it turns into a neoplastic or and malignant cell. As far as the DNA is concerned, the nitrosamines, benzenes, aflatoxin, etc., they are the examples. Radiation, the most common example is atomic explosion, particularly in Japan in the World War, Hiroshima and Nagasaki. Viruses are very important, and one of the example is HPV or human papilloma virus. Chronic inflammatory states, chronic osteomyelitis is very common, and it has been seen that these chronic inflammatory states, they may change the structure or they may damage the DNA or the cell regulatory cycle and produce neoplasia and or malignancy. Some peoples, they are predisposed to the development of cancer. The most common example is CA test. This predisposition runs in families. The most common is carcinoma of the breast. The others are Fanconi's anemia, Bloom syndrome, etc. The carcinogens and carcinogenesis. Carcinogen is a substance which is capable of directly causing cancer in the living tissues. The process by which the carcinogen produces cancer is called carcinogenesis. It is also called oncogenesis or tumorogenesis. Oncogenes. Oncogene basically they were normal genes, but when some of the damages occurred on the DNA or some other parts, these genes become abnormal. These genes change, they become mutated, and now they are capable of producing neoplasia. The process by which normal cells are transformed into cancer cells is called carcinogenesis. There is initiation of cancer formation in the process of carcinogenesis. The process is characterized by changes at the cellular, genetic, and epigenetic levels and at the abnormal cell division. The cell division, it also becomes abnormal. Pro-carcinogen, just see the name. Pro-carcinogen, that is the step before the production of a carcinogen or a malignant disease producing element. Pro-carcinogens itself are not carcinogenic, but after metabolic activation in the body becomes a carcinogen, having a direct genotoxic effect on the DNA. Cytochrome P450 is a very important enzyme system and is present in most of the organs, and it has a metabolizing effect on the different substances. What are the effects? Normally, they cause deactivation of most endogenous compounds and xenobiotics, which may be dangerous for the body or harmful for the body. But at the same time, they may also cause oxidative activation of a pro-carcinogen to carcinogen. So this is how a pro-carcinogen which has been ingested or which has entered the body is converted to a carcinogen by the system of cytochrome P450. These carcinogens then bind to DNA, leading to gene damage and mutation. Mutation then change. Now the gene is changed from a normal gene to a cancer-producing gene. Examples are in the fossil fuels, in the animal fat and smoked meat. There are substances, chemical substances, which are called polycyclic aromatic hydrocarbons. They act as pro-carcinogens. In tobacco, especially cigarette smoking, there is a substance called benzopyrene, which causes lung cancer. It is also a pro-carcinogen. In rubber and aniline dye industries, aromatic amines and azo-dyes are used, and it has been seen that they act as pro-carcinogen in producing the carcinoma of the urinary bladder. Tumor promoters. These tumor promoters, they are substances that enhance tumorigenicity when administered after a carcinogen. It is a substance that usually does not induce a carcinogenic response by itself, but that results in a carcinogenic response when applied in multiple doses following a single subcarcinogenic dose of an initiator. So first, a low dose of carcinogen is applied or it enters the body. This does not cause the production of cancer, but it predisposes to the formation of cancer. So these promoters, if they are applied multiple times, then this carcinogen become actual carcinogen and it causes the production of or the initiation of a cancer. Tumor promoters are generally believed to act by affecting the rate of division, terminal differentiation, or death of tumor precursor cells. For example, asbestosis, which is probably a promoter with respect to lung cancer, and an initiator with respect to mesothelioma in the humans, and certain hormones also. Category of carcinogens: Category 1, Category 2, and Category 3. Category 1 are substances known to be carcinogenic for the humans. Category 2, substances that should be regarded as if they were carcinogenic to the humans. Category 3, substances that have possible carcinogenic effect in humans but about which there is insufficient information to make an assessment. So the category 1, they are known substances. The category 2, they are not so much known and it is thought that they are carcinogenics for the humans. Normal regulatory genes that are principal targets of the carcinogens. Growth promoting proto-oncogenes. So growth promoting genes, they are the targets of for the production of cancer. Growth inhibiting tumor suppressor genes, they are also a target and their abnormality induces malignancy. Genes regulating programmed cell death, which is called apoptosis. Apoptosis, as you know, is a process, is a programmed cell death. Every cell has a definite lifespan, and when that lifespan is complete, then the cell dies by itself. Genes involved in DNA repair causes genetic instability. So if there is abnormality in these genes, the DNA which is damaged does not repair and causes gene mutation. Now, growth promoting gene, which is also called proto-oncogenes, gain of function. Now, more and more cells are produced. Abnormal cell and more cells are produced because the function of these genes is to proliferate as far as the cells are concerned. So now the negative feedback mechanism is inhibited and now there is more and more production of the specific cells. The second one, loss of function. Loss of function is for the growth inhibiting tumor suppressor genes. Whenever a tumor tries to arise, these genes they suppress the tumor, and if they become functionless, then the tumor will not be suppressed and it may develop to the clinical levels. Loss of function, gene regulating program cell death. That is, initially these genes are responsible for the apoptosis, but when they become abnormal, they don't cause apoptosis and there is accumulation of the concerned cells. So, initially there was a problem in the growth promoting oncogene, then growth inhibiting tumor suppressor gene, then genes regulating program cell death. Then at the end, genes involved in DNA repair causes genetic instability, a loss of function. Cell acquires mutations at an accelerated rate because the DNA is not repaired. It becomes abnormal. It is unstable, and when it occurs, then there is a chances that cancer can be produced. Fossil fuel examples: coal, oil, kerosene, propane, natural gas, coal जिसको हम कोयला कहते हैं, oil, पेट्रोल हो गया, केरोसिन मट्टी का तेल, प्रोपेन ये सिलेंडर्स में जो गैस चलती है, नेचुरल गैस जिसको हम पाकिस्तान में सुई गैस भी कहते हैं. Epidemiological factors for cancers, they are environmental, acquired predisposing conditions, genetic predisposition and interaction between environmental and inherited factors. Environmental infectious agents, smoking, alcohol, diet, obesity, reproductive history, environmental carcinogens. Acquired predisposing conditions, chronic inflammation, precursor lesions, immunodeficiency states. Genetic predisposition, breast carcinoma, BRCA1 and BRCA2 genes. They are mutated tumor suppressor genes and reproductive history. Lung carcinoma, polymorphism in cytochrome P450 enzymes metabolizes pro-carcinogens to active carcinogen. Chemical carcinogens, the topic. The first column, we have got names: arsenic and its compounds, asbestosis, benzene, nitrosamine, nickel, radium, hexavalent chromium, etc. etc. Now, the cancer which are associated with these carcinogens: for the arsenic, lung cancer, skin cancer, hemangiosarcoma, and when they are used for occupational use, for example, in herbicides, fungicides, animal tips, drinking water from the contaminated alloys, electrical and semiconductor devices. So these are the occupational hazards in which the person is exposed to arsenic, and this exposure causes may cause lung cancer, skin cancer, and hemangiosarcoma. Asbestosis, occupational use, for example, in the construction industry, roofing papers, floor tiles, fire-resistant textiles, and brake pads. They cause lung cancer, GIT cancer, pleural and peritoneal mesothelioma. Benzene. Benzenes, they are used as light fuel oil, former use as solvent, commodity chemical, and they produce leukemia and Hodgkin lymphoma, and so on. There are a lot of things. Nitrosamine, cigarette smoke, nitrite treated food, they cause lung cancer, esophagus, and liver cancer. And so a lot of these things are there. Bad chemicals. Now you can see the top. The top row contains carcinogens, pro-carcinogens, and tumor promoters. As far as the carcinogens are concerned, they are arsenic and its compound, benzene, nitrosamine, hexavalent chromium, radium, and plutonium. They are all carcinogens, examples of carcinogens. The pro-carcinogens, like hormones like diethyl bestrol, it produces breast cancer. Aflatoxins, it's a fungal toxin, it produces liver cancer, and so on. Aromatic dyes, benzopyrene. Tumor promoters, chlordane causes liver cancer. DDT, which is a spray for the mosquitoes and flies and domestic insects, it causes breast cancer. Phenobarbital, a drug which is used in epilepsy, may cause liver cancer, and so on. The beta methylflavine causes liver cancer, and so on. These are the examples. Now this is a table which is showing a pathological condition and which causes the neoplasm, and what will be the etiologic agent which is causing this neoplasm. Asbestosis and silicosis causes mesothelioma and lung cancer. The examples are asbestos fibers and silica particles. So a long list, and it is a very small list, but there is a number of agents causing these things. For example, if you see number 9, item number 9, hepatitis. Hepatitis is produced by hepatitis B and or C virus, and it causes hepatocellular carcinoma if not treated. And number 10 is osteomyelitis, and osteomyelitis is caused by bacterial infection, and it can cause cancer in the draining sinuses. The last one is chronic cystitis. Chronic cystitis is caused by schistosomiasis, a parasite most common in Egypt region, and if untreated, it can produce blood cancer. Now, this is a list which is called inherited predisposition to cancers. On the top, in the red, is first autosomal dominant cancer syndromes, and in the center is on the with the red color is autosomal recessive syndromes. And the examples are for the autosomal are seven, but they are more. And one of the common example is retinoblastoma, which is produced by a gene called RB or retinoblastoma gene. So it is a autosomal dominant cancer. Number three, you can see melanoma, a cancer of the skin, the melanocytes, and it is produced by a gene which is called CDKN2A, a difficult name to remember, and so many a lot of sort of cancers. Number six, breast and ovarian tumors, especially in females, and they are due to BRCA1 and BRCA2 genes. Now coming to autosomal recessive syndromes of defective DNA repair, inherited predisposition to a condition called xeroderma pigmentosum. There are diverse genes involved in the gene repair. Many genes are involved in gene repair, and the abnormalities of these genes causes xeroderma pigmentosum. Ataxia telangiectasia. It is an autosomal recessive syndrome due to defective DNA repair, and the gene which becomes abnormal is ATM. And number three is Bloom syndrome and Fanconi's anemia, and you can see the name of the genes which have been transmitted from the parents to the patient. Based on their biological activities, chemical carcinogens can be classified into genotoxic carcinogens, DNA reactive, and non-genotoxic carcinogens, for example, non-DNA reactive or epigenetic. Direct carcinogens activate without metabolic activation, as we have seen earlier. Indirect carcinogens or pro-carcinogens, they become active after metabolic activation, and by which enzyme system? The cytochrome enzyme system. Now coming to non-genotoxic carcinogens, they don't damage DNA directly but alter the way in which DNA expresses itself or expresses its information. They act in other ways to promote abnormal cell growth and replication. They may also cause cell cycle disruption when the cell or tissues is more prone to DNA damage from another source. For example, hormones, some organic compounds. Like for the example, we have seen the promoters. Examples of non-genotoxic carcinogens or promoters of tumor transformation are heavy metals such as arsenic and hormones such as estrogen. Classification of carcinogens. Genotoxic, non-genotoxic, again the same thing, maybe a repetition of the slide. Genotoxic binds to DNA, causes irreversible damage producing mutations. Chemical agents like nitrosa and methylurea, a very difficult name to pronounce, or non-chemical agents such as ultraviolet light and ionizing radiations. Non-genotoxic, they don't damage the DNA directly, but act in other ways to promote growth. So this slide is basically a summary or repetition of the previous slide. The non-genotoxic, they include hormones and some organic compounds. Now, genotoxic carcinogens, which act directly on the DNA. The carcinogens interact physically with the DNA to damage or change its structure. Genotoxins cause irreversible genetic damage and produces mutations by binding to DNA. Genotoxic include chemicals which we have already seen, for example, N-nitrosa and methylurea. Chemotherapy agents are very important. Sometimes we give chemotherapy for a certain malignancy. The malignancy may be cured or becomes better, but these chemotherapy agents may damage the other cell lines and a secondary cancer may emerge. Ultraviolet light and ionizing radiation, for example, in Australia, the exposure to ultraviolet light is very high, and there is a lot of patient with skin cancers. Certain viruses interact with DNA, and this induction is a recognized primary origin of neoplastic transformation. So viruses are also responsible for the DNA damage. And we have seen the example of human papillomavirus in our initial slides. Now the non-genotoxic carcinogens. It is a repetition of the previous slide, but as it is very important, so I thought that we should review it again so that the things get into our minds. These alter the way in which DNA expresses its information. That is, it doesn't damage the DNA, but the information which the DNA gives for further progression of the function, they change these informations. So a misinformation is relayed from a normal gene or normal DNA. They act in other ways to promote abnormal cell growth and replication. They may also cause cell cycle disruption when the cells or tissues are more prone to DNA damage from another source, such as hormones and some organic compounds. These hormones are frequently used in the facial creams and in some other parts of the body. So they should only be used under the supervision of a qualified medical doctor who has specialization for such sort of hormone usage. Examples of non-genotoxic carcinogens or promoters of tumor transformation include heavy metals such as arsenic and hormones such as estrogen. This is a female hormone. Exposure to carcinogenic agents is associated with various forms of DNA damage. Single strand breaks, double strand breaks, covalently bound chemical DNA adducts, oxidative induced lesions, DNA or DNA protein cause links, a difficult slide. And I think we should only remember the single strand breaks or a double strand breaks. The rest they look difficult to me. Genetic lesion in the cancer. They may range from point mutation in a single nucleotide to large abnormalities large enough to produce gross changes in the chromosome. They are the mutations, gene rearrangements, deletions, and epigenetic modification and cancer. As far as the mutations are concerned, they are driver pathogenic mutations. They are usually acquired. They change the function of the oncogene directly contributing to development or progression of a given cancer. They may be passengers also, neutral, and they are acquired from, for example, from some of the viruses. Gene rearrangement, translocations, that is a genetic part from one chromosome breaks, and at the same time some part of the another chromosome also breaks and they change the material, so an abnormal gene is formed which is called translocation. Trans means across, location means location, so the location becomes across to another chromosome. These are translocations. Inversion, for example, one part of the chromosome breaks and instead of rejoining at the same point, it inverts itself upside down, and the down portion or the down part attaches to the broken part. So these are called inversions. So now the gene structure has changed. A new novel gene is formed, which, if abnormal, can produce a genetic lesion, and that genetic lesion can cause cancer. Deletions usually affect tumor suppressor gene. If the part of the chromosome which harbors tumor suppressor gene is broken and lost, then the effect of the tumor suppressor gene will not be there, and the tumor which were being controlled by these genes, they will not be controlled, and now there will be progression of these malignant cells to produce a full-fledged tumor. Gene amplification increases the expression of oncogene. Epigenetic modification in cancer. No mutation of DNA or genes, but may affect expression of tumor suppressor and or DNA repair genes. So what are epigenetic modifications? There is no effect on the DNA or the genes, but the expression of the tumor suppressor or the DNA repair genes may become abnormal. Events in carcinogenesis. Proto-oncogenes are normal genes, as we have already discussed. They become oncogenes due to DNA damage and mutations. So now that normal gene, which was called proto-oncogene, now it has become a malignant oncogene due to mutation, due to a damage in the DNA system. Mutation in a proto-oncogene can generate an abnormal clone cell population with a distinct advantage in proliferation. Events leading to development of cancer broadly divided into the stages. The first stage is initiation. The carcinogen produces mutation. Cells become cancers or cancerous cell or cancer producing cell. Promotion. The cancer cell multiplies more and more and more and more. Progression. More mutation occurs. Initially, there was one mutation, but due to the instability of the DNA or the gene, more mutation have occurred and cancer becomes more aggressive, which may occur over a long period of time depending upon the chronic and extent of exposure to the carcinogens. So there are three stages in the development of carcinogenesis or in the process of carcinogenesis: initiation, promotion, and progression. And these all are due to the formation of a proto-oncogene to a malignant oncogene due to DNA damage which causes mutation. Steps in cancer formation. First hit occurs to DNA gene by a carcinogen, but it looks like that it was not enough. A damage occurs to a normal regulatory gene or its product. This DNA has to be repaired. Repair of the DNA occurs via genes involved in DNA repair, which we have already discussed. If the repair is done promptly and adequately, then function returns to normal and nothing and no abnormality occurs. But if the DNA repair is not done due to malfunctioning of the genes due to its mutation, that is, there is a mutation in the genes causing repair function. If they are abnormal, then this DNA damage remains there and it is not repaired. Multiple new hits or damages occurs to already mutated genes over time. Ultimately, normal regulatory control of cell proliferation is lost and a cancer has emerged. So these are the steps in the cancer formation. The first hit doesn't necessarily cause a cancer, but other abnormalities, if they are present, they may cause or they may initiate the formation of a cancer. Hallmarks of cancer. Self-sufficiency in growth signals. They are self-sufficient. They are not dependent upon the body or the normal cycle. They are outlaws. They don't believe in any law. They don't follow the law. Insensitive of growth inhibitory signals. The signals, normal growth inhibitory signals which are produced, these cells, they don't care about them. They are not sensitive. They keep on producing their malignant cells. Altered cellular metabolism. Their cellular metabolism is changed from a normal to an abnormal one. Evasion of apoptosis. Now they don't die. The programmed cell death mechanism is lost now. So the cells they don't die early. They have a long life. Immortality. Due to this long life, they become immortal. They don't die. Sustained angiogenesis. In order to remain alive, these cancerous cells they need food products. These food products they are provided from the blood vessel. So they have a capability that they produce angiogenesis or production of new blood vessel. So the raw material for their growth can be obtained from it. Invasion and metastasis. Invasion is they directly invade some of the organs like liver, like spleen, or like brain, or whatever it may be. And metastasis, that is, they are present at one place, but they go through different vehicles like blood supply, like lymph flow to other organs remote from the area of the origin. These are called metastasis. Evasion of immune surveillance. Usually, the normal immune surveillance is a system in which any cancerous cells which is produced, our immune system it kills that cell. But now these cells, malignant cells, they have developed the how to bypass this immune surveillance. So they are not killed by or they are not removed by immune surveillance mechanism which is present normally in the body. These are the examples which can produce cancer. They are all delicious dishes. There are burgers and there are French fried chips and there is pizza also. And in the right one, in the corner, you can see either it is Pepsi or Coke. Then on the right side, there is barbecue. There is lamb chops or some chicken sort of thing and some kebabs also. In the bottom, I don't know what is this things called, but they are eaten frequently by the people. And the cancer causing food again the same burgers. It contains kebab, I think, which is smoked kebab. That is why it is causing the French fries, the oily foods, and all those things. They all the causes cancer. They predispose you to cancer, not necessarily causes, but they may cause damage to the DNA system or the normal regulatory cycle of the cells. They may damage it. So better to avoid them, but it is difficult to avoid them. Now these are the occupational hazard. The person is spraying some pesticides for some, unwanted grass or something like this, and at the same time you can see the chimneys. A lot of smoke is coming and very bad carcinogens or promoters or whatever they may be in it, and it is polluting the whole environment. On the right side you can see a person is smoking, and this smoking, it causes lung cancer and it is very dangerous. It's also called COPD, like the lung disease also, but the most dangerous one is the lung cancer. These are difficult things, but you just have a look at them. Single strand break, double strand break, and all these things which are happening to the DNA. And these carcinogens, they are producing these things. So our brain has to have spectacles, and we should realize that what we are eating and in which environment we are being exposed. For example, in an environment in which there is a lot of ultraviolet exposure, for example in Pakistan, especially in the coastal areas in summer season, the ultraviolet exposure is very very high, the index is very high. So we have to be careful. If we are walking under the sun directly, we have to cover ourselves. And some people also recommend skin blocking agent also. Better to avoid, but be covered when we are walking in this at the time when there is high ultraviolet or UV index. Thank you very much. Again the same. Oh, again say the same is come. Oh, so this is all the cycle we have called DNA damage. It is in sort of uh, table form. Multiple carcinogenesis. Again, it will be very easy for you to again the DNA damage. This the slide for the delicious food. It looks very very delicious, and it's got fried chips. It has got pizza also, and a lot of things you can see in the sauce also. I don't know. It looks very delicious. Anyhow, thank you very much. And if you have any questions, please try to contact me. I am available there all the time in the working hours from 8:30 till 3:30. And in Ramzan, they are a bit shorter. So you can contact me. You know my office, and you can contact me anytime. But please, before coming, please do contact me, whether I am there or not, because I have other works in the diagnostic also. So I may not be available at that time, and then we can have a time and then we can discuss the things. Thank you very much.